my goals for today are to kind of get people thinking deeply about what ecological monitoring is, what your research questions might be. I think you've identified some of them. But then more specifically, how do you take those research questions and think about the nuances of those research questions and various facets of those research questions at different scales, both in space and time. And then given that more critical thinking about the research questions you're hoping to answer with your monitoring, and that might be monitoring being done by scientists and or students in your classrooms, and it might be monitoring collectively across all of your programs. Maybe you're doing concerted, consistent, standardized monitoring across multiple classrooms, for example. That might be an alternative. But once you've given some critical thinking to the scale at which your monitoring is happening and the more nuanced, specific research questions you hope to answer, how do you then have folks, either students or professional scientists or in tandem, both of those people, collect rigorous monitoring data using citsci.org tools and or other tools that you might have at your ready, to collect rigorous scientific data that is really going to help you answer those research questions. So kind of in general, we're talking about monitoring protocols and potential use of citsci.org to create and identify and determine those protocols. And when we talk about ecological monitoring, let me put this into a full screen for you guys. And let me know if that's too big, and if you can't see it, chime in. But some important questions at the very beginning of ecological monitoring is kind of just that: what are you monitoring, and for whom are you monitoring? That is it an agency that would like some information, like the U.S. Forest Service? Is it or is it a scientist you're working with in tandem and in partnership with? Is it yourself as a class, etc. And so. And then for whom and to answer what questions. And welcome Sarah. It looks like Sarah Urban just arrived, so welcome. So what are you trying to monitor? For whom are you monitoring that ecological system? And to answer which questions and at what spatial and temporal scales and how. So they're kind of broad, high-level questions, sometimes hard to answer. But I had given this slide in my previous webinar, and I'm going to revisit it, because if you can think of your classroom or the BANR project, a team of individuals keeping track of something and monitoring it, you can think about it through time, and you can think about it through space, and you can look at maybe trend analysis. Like in this case, you might have an intervention, maybe a treatment, a fuels treatment or something, and you might want to look at the effect of that intervention on the variable that you're measuring. In this case, it's kind of hypothetical, but like woody biomass or beetle kill trees, number of trees that are infested with beetle kill, for example. And so this is just a real quick visual of what monitoring can look like at one scale. For maybe two variables given an intervention. But so how do we do this? And in many cases, we can think about it as defining our question, gathering information, developing explanations, choosing methods, collecting data, actually going out in the field and boots on the ground, collecting data, and then exploring and analyzing and interpreting that data online once collected and shared across groups, collecting the same data, for example, and then sharing results and evaluating success. But I'm going to open up with some questions that, this is all entirely kudos to Beth, but she gave and reiterated a few questions that I thought I might start with. And this may be familiar to you guys, but there might be a question looking at relationships among different systems, and we might consider those as macroscopic scale questions, such as does adding biochar to soil help plants grow? And of course, we can devise some experiments to answer that question. But to think more critically about that question, we can look at mechanisms about how does adding biochar help plants grow. We also can think of different subsystems related to that question, such as roots and cells and structures of biochar, etc., and consider and ask a question: what properties and processes at this smaller scale may lead to differences in plant growth? So this hopefully is familiar with you guys. And then at a larger scale, at the opposite end of the spectrum, we can look at questions about the significance and emergent properties of the treatments that we're conducting, like the addition of biochar, for example. And you can ask questions like, how does biochar affect ecological matter cycling and energy flow? And why do we care about these effects on plant growth? And maybe even at a larger system, what is the effect of these additions at the larger scale across farms and watersheds? So basically, we're moving from various levels of scale, and I think it's important to keep these things in mind as we go forward. So let's see if I can't go to the next. So some example questions again: what will the ecological environmental impacts be for different forest treatments, for example, in an area with beetle kill? Maybe removing beetle kill versus leaving it standing, and where, when, and how harvesting wood, especially but not solely beetle kill wood, for bioenergy uses might be environmentally appropriate or economically feasible. So although these sound great on paper, how do you actually translate these questions? So here's a couple different scaled questions. Maybe you have a forest inventory that you are looking at. Maybe you have questions about soil. Maybe you look at species patterns across the large forest landscape across the entire Intermountain West, for example. So despite these questions, how do you then take those on paper, which sound really good regardless of the scale they're at, and actually implement something in the field and actually collect data and devise experimental designs and data sheets and actually go about implementing that? So as a review, very short, I'm going to kind of go into citsci.org in a little detail, and then hopefully what I hope to do is together have us walk through how to figure out how to devise a data sheet and a protocol and an experimental design that might answer a variety of different scaled questions. As a reminder, thanks to all for telling me you've been introduced, of course, to citsci.org, but it's a free open online support platform supporting citizen science programs. It provides tools for project and data management, and it aims to support the entire citizen science process, from creating projects to managing members, such as like approving your students as members of a project, and therefore they have permission to enter data. That data might be the diameter of a tree, or it might be a treatment that was, maybe you're documenting that this particular plot had been burned or something to that effect. So you can create projects, of course, build data sheets so that you can enter the type of data you want your volunteers to measure, design protocols, explore data, visualize data, document and share data, and evaluate success. And so I put this slide up here just to remind us that when you're creating projects, you're making these very hard decisions, such as what your research question is, and how you're going to measure certain things at certain places at certain times to answer those research questions. So that there's really project design considerations to consider. And some of those considerations include devising experimental designs. And these designs might be anything from, and so for example on citsci.org there's the Urban Ecology Center in Milwaukee, Wisconsin, that is doing snakeboard monitoring transects, and they created this, they came up with this, they devised this this hexagonal transect scheme, and what they did is they ran transects, 1, 2, 3 of them, across the edges of the, from edge to edge of this hexagon, and along each transect at every, I think, 5 meters, they put down a 1 by 1 meter piece of plywood, and they say, okay, we're going to have our classroom citizen scientists, we're going to have them measure and count the number of snakes by species they encounter when they lift the plywood up at each of the plots along that transect. And they do that on transect 1, transect 2, and transect 3, and then they repeat this at a different hexagon at a different location in the meadow or field in Wisconsin. And so that's one of many examples. Other examples are the infamous Daubenmire transect, many of you may be familiar with, where you walk down a transect and you plop down 1 square meter actually they're smaller than that now, little rectangular PVC frames, and you look at percent cover of invasive plants, for example, in each frame along a transect. There's a simple point observation for species distribution mapping and modeling and that type of work. So you might use a point to say, at this location I found a red fox, for example, and maybe you're interested in, is the encounters of red fox significantly different in ponderosa pine forest versus lodgepole pine forest, for example. And so you might use opportunistic point observations to begin to think about that question. There's the U.S. Forest Service Forest Health Monitoring plots, and they're circular in radius, and they consist of three, what's in, I think in this picture, maroon subplots where you do understory vegetation, and then you do overstory vegetation in the blue outer circle. And this is a traditional, what they call forest inventory or analysis, or FIA plot, that the Forest Service uses for forest health condition monitoring. Other survey types might be political boundaries, such as a natural area in your neck of the woods, where you maybe conduct a bioblitz, for example. And then, as my background is in plant ecology, you might use a very sophisticated modified Whittaker plot to look at species accumulation curves and determine how adequately you've sampled a national park, for example, to look at plant biodiversity. And it looks like we have a question in the chat. Thank you, Mary. And your question is, based on your experience with classrooms, how much are students involved in designing projects and designing protocols? It's a great segue and a great question, Mary. And I think it depends on your learning objectives. To some degree. It also depends on the scientist partnership liaison you have formed with, say, BANR scientists to determine how sorry, I had a phone call come in on another call here, so I got distracted. So back to your question, Mary. It depends somewhat on the scientists' objectives as well. So in the BANR project, you could certainly ask your scientists what flexibility you have in having your classroom be engaged in designing their own protocols, and how that might dovetail with the science objectives of the project. That would be one thing to do, is talk with your partner scientist there. But in my experience, I've found that it can be a great learning opportunity for students to think about designing protocols, and it's very powerful to ask them, if this is your question, how would you measure it? And so I feel like if nothing else, it's worth a discussion in the classroom to have students think critically about sampling design and designing their own projects. So hopefully that answered your question. It's a great question. So once you've decided about, kind of worked on your experimental design, and you not only thought about what plot types you're going to use, but then you also thought about how to just lay out your plots on the landscape and at what scale on the landscape is appropriate to your research question. So for example, in this modified Whittaker plot, what we tend to do as plant ecologists is we've chosen this design for a very, very targeted reason, and that might be to measure the number of plants found in each of the blue quadrats along the perimeter of this plot, and then those are 1 square meter, and so we can say on average we encountered 10 plants in that plot. And then if you measure how many plants you found in what's called A and B on this slide, on the right there, those are 10 square meter plots. Maybe you encountered maybe 12 plants. And then you search the entire area of C, which is 100 square meters in size, and maybe you came across 13, and then maybe you did a full search of this full rectangle, and you still encountered only 13 different species. If you did that, you end up plotting by area the number, so your y-axis becomes the number and your x-axis becomes the area at 1, 10, 100, and 1,000 square meters, and you have this curve that levels off. And if it does that, then that indicates to you that you've encountered as many plants in this habitat type as possible, so long as you've identified and put these modified Whittaker plots out, say 10 of them in what they call a stratified design. So you stratify by habitat type. So you might put 10 plots in lodgepole and another 10 plots in subalpine fir and another 10 plots in aspen forests. And if you did that, you could then experimentally ask which forest type has the most plant diversity, understory plant diversity, for example. So that's an example of not only the experimental design, the plot design, but also how you lay out those plots on the landscape. And then, of course, choosing what units to measure. Are you measuring number of plants? Are you measuring, height? And if you're measuring height, is it in centimeters? Is it in meters? These are all things that are, need to be answered. And then, in some cases, monitoring organizations, in this case Trout Unlimited in Pennsylvania, happens to be measuring stream pH and stream total dissolved solids and the connectivity of the water at the same location many times. So they're doing repeat monitoring throughout time, so their scale is time, and they're looking at changes in pH and total dissolved solids, for example, through time. And they're doing that because they want to identify the impact of, say, fracking and say, okay, throughout time, after the onset of fracking, the pH decreased, or for example. Other plot or monitoring schemes measure many locations only once. So this is the Front Range Pika project here in Colorado, measuring American pika populations, and they're opportunistically trying to get a spatial view, not over time, but over space, of where American pika populations are statewide, so they could develop a distribution of that species. So at the end of the day, regardless of whether you're measuring one place consistently over time or many places at one time, most projects create data sheets. And what our experience is when they create their data sheet, they go into Word and they come up with this great little design of measuring, say, water temperature and clarity, but they have a hard time translating that into digital data and thinking about what that data might do and how they want to use that data, kind of like a begin with the end in mind, to say, how do you, once you get this data, what type of format will it be in what type of spreadsheet or database, and then how will it answer the question that I set out to answer. So how do you go from these types of data sheets, these monitoring data sheets that I've posted up here, to a database? And then also remember that some projects, let's say you're Doug in Wyoming, and let's say you're another teacher here in Colorado, you might choose to measure the same thing in the same way. Or you have a unique special project that's specific to your location and your question, and you might measure things in very different, unique ways. And so regardless of approach, citsci.org is hopefully a system that might help you do this. And the way it does it is through its datasheet creator, which I'm going to talk about in detail a little bit later as we get into this system. But in this datasheet creator, you can specifically say these are opportunistic locations, kind of like those points, and it says entered by user here in the middle of the screen, or predefined. And so what this allows you to do is actually think very critically about, are you like that Trout Unlimited group that's going to the same location over time, or are you opportunistically measuring like pika, for example? And so that's an option you have to choose from. And then this observation type lets you pick from different transects. So like that snakeboard monitoring plot would show up here. In this case, it's a simple point observation, but we can help you create, if you've come up with a design, we can help you create that design in the database in the back end, and it would be selectable, and you could pick, like I went to a BANR forest monitoring plot if that's what you've devised. And so in that case, your students could be asked to just pick that, well, go to the data sheet and enter data. So here you would say, this is the type of plot I'm going to use for my protocol. And then at that plot, what species, so add organism, what maybe you have a pick list of organisms. You know that there's like 10 species you might encounter, and so you want to keep it really easy for your citizen scientists to, pick a species from that pick list. Or you might want to add an organism pick list for any organism, like a BioBlitz, or you might want to measure a site characteristic, such as like the percent sand, or the pH of a system, or that kind of thing. So if you then say predefined, you'd be able to enter plot 1 and plot 2 and specify its locations as a project coordinator, and you'd be able to enter your different data sheets the way you need it. So basically, you can collaboratively define measurements that you'd like students to measure. They can be numeric, they can be integers, decimals, they can be categorical data, so they could be like a subjective, like soil texture. It might be sandy, it might be sandy clay loam, it might be loamy, for example, and you can create your own categories as needed for that categorical variable. You can have your students enter text, straight up text, to describe, say, the disease, level of disease, or the disease evidence of disease for a tree, for example. And then you can have them enter multimedia, such as photos, take pictures of the plots at that location for, say, repeat photography, for example. So as many of you might have heard in a previous webinar, basically this is what's being measured. It's an active growing list of what different teachers or citizen science project coordinators or researchers are measuring, and you can certainly pick from that list, that growing list of things being measured, or you can create your own new measurement to meet the specific needs of your monitoring protocol. So in this case, you can define of course, as I mentioned, your own monitoring locations if need be. And then after the data come in, back to that Trout Unlimited volunteer example, you can get dynamic visualizations and data and explore the data that your citizen scientists, students, or participants have collected. And so in this case, you've selected air temperature because that's something you wanted your volunteers to measure, and they've measured it since 2013, and you can look at trends in air temperature at this specific location through time. The system will automatically create a frequency distribution of the variable that you've selected in this pick list, and it'll give you some summary descriptive statistics, and these are dynamically updated as new data are entered by students or volunteers. So additionally, you can look at comparisons of different plots. So in this case, this is, again, Trout Unlimited monitoring data, looking at this plot called site Alaria 001 and like Babb Creek, BABBCR 001, and you can see that its particular air temperature on average throughout time has been lower perhaps than Babb Creek 2, for example. So this is a way to explore your data, kind of investigate trends that you might be seeing in your monitoring data. So with that introduction, I'm going to delve into actually kind of talking with you guys together about some of the schemes you might be monitoring and some of the research questions you're hoping to answer, and I'll bring up citsci.org as an example so that we can get into the system. So bear with me here, and I am going to log in, and I'm going to pull up a project that's just barely getting started, and I'm a manager of that project. And so because I'm a manager, I have some abilities to edit data sheets. And this is the Forest Health Task Force here in Summit County, Colorado, or Summit County Forest Health Task Force. And it's just getting started, so there's not much data in this project yet, but it'll be a great place for us to look at different data sheets and create a data sheet and think about how you as a project might be able to construct and devise and design a protocol for your citizen scientists to work on. So I'm going to create a new data sheet so that we can work on a new data sheet here on this project, and I'm going to click New Data Sheet, and it's a little slow in loading today, so bear with us. So what this is going to do is it's going to bring up that data sheet creator, and it should come up pretty quickly. I might pull up a different test website too because it's somewhat of a busy site right now, so bear with me. Okay, so here's our datasheet creator.
Can you make
And this is
can you
kind
make
of
the
the,
screen a little bigger?
sure. Is that big enough or do you need bigger?
It was Mary who was asking, so.
Let me see if I can't zoom in here. And is that better?
It looks better. Thank
Okay,
you.
we'll go with that. Oh, great. Mary says it's looking a little better. So
It's
this
much
is,
better. Yep.
oh, great. Thanks, John. So this is kind of where all kind of protocols are kind of conceived. And so, like we were talking about earlier in the webinar, this is where you need to make some very critical decisions about your sampling design, your experimental design, your plot type, and that kind of thing. You can name your data sheet. So this might be Forest Biochar Monitoring Data Sheet. So this is a hypothetical data sheet, and you might give your students instructions. So please, let's see here. Please remember, just notes for your students to make note of. Please remember to measure, I don't know, tree height in feet. Maybe you actually want them to measure it in feet, or maybe you want to have them measure it in meters. It's just something to remember, or bring these instruments to the field with you. This is just a nice little place to give them instructions on how to fill this data out. And as we were talking about, you can have predefined monitoring locations, or you can have the location entered by the user. And what that means is if a student were going out and instructed to go out behind a school and say measures, say, a tree, they could actually say, well, where did they measure that tree, and they could use a map to identify their location. Alternatively, you as a teacher or project coordinator could predefine and assign students certain locations for monitoring. And this might work quite well in the case of forest health monitoring. So I'm going to choose predefined locations, and I'm going to click add location, and I'm going to call it plot 1 as an example. And I'm going to zoom in to, I don't know, Summit County. See if it knows that Colorado, or maybe Frisco, Colorado. How about we do that? So that's a town, and you can search by that name, and sure enough, it found Frisco, Colorado. So let's say that we know that we have a high school around here, and we know that there's a monitoring plot right over here by this creek. So you can find your location, and it'll automatically know your latitude and longitude for plot 1, and you can hit submit. So, and it looks like the name already exists, so I'm going to call it F Forest Health Monitoring Plot 1. So I'm going to hit submit. And so now I've got Forest Health Monitoring Plot 1. I can add a second location. It will remember where I was before. And I locate, but maybe I'm over by this creek. So now I'm at what they call a Forest Health Monitoring Plot 2. And I click Submit. And so now I've got 2 predefined locations through which I could assign to my students in my class for them to go to make measurements. And so now the next question in devising your protocol is to determine what measurements you'd like them to make. And as Beth had mentioned in the slide, I kind of reiterated she had given earlier, you have to decide what scale your measurement is being made. So is it a very fine scale and you're measuring soil because you're looking at carbon cycling, or not like carbon cycling, but let's say you're looking at soil texture or soil respiration, or is it a bigger measurement like average forest biomass by species, for example, or average tree diameter by species, which could then be aggregated at the landscape scale by someone in the U.S. Forest Service, for example, or a BANR researcher to look at maybe trends regionally across the entire Intermountain West for like forest, tree diameter trends by species, for example. So remember to keep those kind of scale questions in mind when you're devising your protocol. But in this case, let's say we want to measure the diameter of a particular tree. So we could type in lodgepole pine, for example, and see it's going to go search. As I type, it's automatically going to search, and this can take some time because it's looking through all plants that we are aware of. But you only have to do this once, and you only have to devise your data sheet once. So here we go. Here's Pinus contorta. So I click select, And what this will do is bring that tree into my list of organisms or species I'd like my volunteers or students to measure. So now you have to ask, what measurement do you want your students to measure about that tree? So you can click Add Measurement, and you can select height, for example. And turns out you can just select it, and now you've got the height. Well, next question is, what units would you like your students to measure the height in? So you can click units, and you can say centimeters or meters or feet. I think we had said meters in our instructions. So now you're measuring or asking your volunteers to measure the height of an individual tree in meters. You might also add a second measurement, and that might be diameter at breast height, for example, and you can say select it. Now you might also, and this might be in, I don't know, centimeters. So you might also say, well, what if I want to measure evidence of disease or something? Or like the number of, I don't know, numbers of spots of evidence of beetle kill on the tree. And it might not be in this list. So you might search through this list of all things being measured, tree condition, for example, and you might say, you know, I don't see what I need, so what do I do? So let's say we will actually want to measure, I don't know, the number of black spots on the fourth leaf. It sounds somewhat arbitrary, but let's go ahead and add it. So if you search for it and you don't see anything called the number of spots, then you're not seeing anything that's being measured already by an existing project. So what you can do is you can say add new measurement. And what this will do is let you say number of spots on fourth leaf. And it's just kind of an arbitrary example, but that might be something you might want them to measure. And so then you can describe the measurement as needed. And you can say it's an individual tree that you're measuring, and it is numeric, and it's a decimal. Well, it's an integer. It's actually the number. And if, for example, you're measuring something that has a min and max range for quality assurance, quality control, like pH, for example, 0 to 14, you can enter optionally a minimum and maximum value. So if you hit submit, you've now got not only the height of the lodgepole, the diameter at breast height, but also the number of spots on the fourth leaf. And this can be unitless, that's okay. Because it's just a count. And so then if you want to measure a site characteristic at this location, you could measure, say, air temperature, for example. So now you've got your students measuring attributes about a lodgepole pine, and you've got them measuring air temperature at these different assigned locations, which happen to be the FHM plot 1 and FHM plot 2. So now you're ready to go ahead and validate and save your observations. I guess I might measure a second site characteristic just to illustrate a few more features. So I'll click Add Site Characteristic, and I'll say wind speed. But again, you have to be very critical, think very critically about what it is you're asking your folks to measure, and make sure that the measurements you hope for them to measure answered your research question. So again, not to lose sight of making sure that your protocol you're designing is answering the question you're intending to answer at the scale at which you're intending to answer it. So here we go. We've got categorical data in this case, and it's wind speed. But let's say you want them to measure that first. You can just click up, and then it will move these up and down. And air temperature, we might click degrees centigrade, for example, and now we've got our units specified, and we're ready to go. So you can click save, and it'll say you're saved, you're good, and you go back to your project. So now we go back to this Forest Health Monitoring, Summit County Forest Health Task Force datasheet, and we've got Forest Biochar Monitoring datasheet. It looks like I spelled forest wrong, so no worries, you can always go edit it. So I'll click edit, and I will go in and edit that datasheet name. This might take a little time to load up, so bear with me. And so you can always iteratively build this with, like Mary mentioned in her question earlier, you can actually go through this process with your students to get them thinking very critically about what their data sheet is. So here's our forest biochar monitoring data sheet. I'll go ahead and save it. And then I'll go back. You can preview it, but I'm going to actually take you to what the student would see to enter data for this protocol. So here's this data sheet, and the students would only see these two, the first two buttons. You as a project coordinator would see the edit and the delete and whether what we're doing is we're validating your data sheet. What that means is if you added an organism but didn't measure anything about it, that would be invalid, and so the system's just telling you that these are valid data sheets because everything seems to check out okay. So it's just another quality assurance, quality control procedure we have. But you as a project coordinator see the last two buttons and whether they're valid or not, but only your students, in this case, see the first two buttons. So they come here and instead they just see two rows of buttons, two columns, I guess. And so they can enter data as a member of your project. And when they do, they click enter data. It loads up the data sheet. And here we go. They're going to be asked for the same questions you just asked them to answer.
Do you have to be online to do this? Is there any offline data entry or no?
Good question. Was that Beth?
Yeah.
Yeah, so good question. There is a mobile app for sitsci.org that's in its beta phase. We're currently, in fact, I think the phone call I had come in just a second ago was related to development of our second version of our mobile app. It's on Android and iOS or iPhone. And so some of the features, I'd say 60% of the features I just showed are supported by our current version 1 of our mobile app out in the field, which would mean that you would go out in the field and you could do it offline. You would not need that connectivity to enter this data, and what you see on this data sheet could be entered on your phone, saved, and then when you return to, say, a coffee shop, you could then submit your observation. Does that answer your question, Beth?
Yeah, you could do iPad too.
You could. Yep, because of the iPhone app, it would work on the iPad. I have it on my iPad. But again, it only supports about 60% of what we're just talking about. And we're working hard to release a version 2, which would support 100% of what we're talking about.
Great, thanks.
So as we go through this, so what the student would see is the date defaults to today. This is going to be a growing list of members of your project. So basically think of it as a classroom list of all your students that you've approved or invited via that invite members feature. So you could say select, basically instruct your students to select themselves as the recorder. And comments, everybody really enjoys entering, and very, very important to enter field comments, much like in a Write and Array notebook, some comments about your visit to that location that you're monitoring. So then you pick which plot you were assigned. And so if you were assigned plot 1, it's going to give you a confirmation so that you can say, aha, I actually went to the right spot because this is a visual of where that was in Frisco, Colorado. And sure enough, there's your monitoring plot. So you pick it because that's where you went to conduct your monitoring. And then you enter your height in meters, I don't know, maybe 6 meters, maybe 18 feet, and then the diameter in centimeters, maybe 22 centimeters, and maybe there's 5 spots on that 4th leaf. So really, this is a very flexible system. The harder questions are devising those protocols that we're talking about that are appropriate to your research questions. So although you can add any measurement you want, kind of like in this case we added the number of spots on the 4th leaf, you have to think very critically, and it's a great opportunity, as Mary mentioned earlier, to have your students think very critically as well about what these questions are, how these plots are laid out on the landscape, how they stratified by forest type, for example, and those types of scale-based questions. So if I proceed to finish entering this data, I maybe, this was a categorical variable. It was a slight breeze. The air temperature in degrees C, I don't know, was 25 or whatever. And then you can upload photos for this plot, so you can browse to your computer. If this is after the monitoring, if it's on the app, you could take a picture of right there as you're at that plot. So then you hit submit, and the data go into the system, and we let it crunch the numbers, and the data show up as a confirmation page. And so this is great for students hoping to see confirmation of their monitoring for that day. It will eventually load up a map of the site, so it's giving you again that confirmation. And that's weird, interesting. And so then it'll give you the ability to edit stuff. So if you forgot to enter comments, you click edit, and it will go and let you add those. It'll also, since you're the person who entered this data, it will let you edit your value from 6 to 7 because you made a mistake. And this is only available for you, the person who entered the data, as well as the project coordinator. So then you're good to go, and you go back to your project profile, and you can submit another observation. So that makes sense. So now what you'll see in the View Data tab is our March 5th today observation at Forest Health Monitoring Plot 1, and you can go back, and this View button will show you that exact same confirmation page. And I was looking at the map and I was a little confused, but now that I look at it, it was just zoomed out. So if we go to the view, we might see the same data. We'll see that 7 that I edited. And so now it's going to, if I zoom in, we'll see that we were in Frisco. Yep, sure enough. So we're good to go. And so that's how, from start to finish, you constructed for this project the Summit County Forest Health Task Force. You created a data sheet, and then you had a volunteer or trained citizen scientist or student enter that data using that data sheet. So at this point, we're about 10 minutes away from kind of concluding. I'll open it up to any questions anyone has, and yeah, we'll go from there. So if you don't forget to unmute yourself if you have any questions. There's quite a few of us who have joined. Let me know if you've got any thoughts or questions about it. I guess the biggest thing to keep in mind that Beth and I were talking about earlier is that to not underestimate the challenge of devising a protocol and a monitoring scheme appropriate to your research question that's appropriate to the scale of your research question.
I do have one question. This is Doug.
Yeah.
Say we devise a project that maybe wouldn't be necessarily accessible to the public or very frequently accessed by the public. Does the project become inactive after a while? Or can they, or is it on there indefinitely forever?
Great question. As a project coordinator, you can edit your project profile, and in this project profile is a field that is status, and this project is starting. You could be in progress or complete. So as the project progresses through time and concludes, you could actually switch that status to complete. Does that answer your question?
That does, because I think one of the ideas that I mean, if I were to do this, right now we go and we collect snow data from a snow fence that's north of our town.
Okay.
We just do that once a year in March.
Uh-huh.
So we take a sample of the snow drift and we do this snow water equivalent of each of the layers of the snow drift. And right now we're just keeping track of that in Google Docs.
Gotcha. What's nice about, so that's perfect. That's a great example of, you could use that add new measurement. So in the what's being measured, or when you're actually building your data sheet, you could add the snow water equivalent, the SWE, of your snow drift as a measurement if it's not in the system already. And what's nice about this is the monitoring frequency is not specified necessarily by citsci.org. So that's, as a coordinator, you could certainly specify the frequency that you measure that variable throughout time. And what would be quite interesting, right, from especially a soil moisture perspective, which has very big implications ecologically, you could track and monitor and visualize snow water equivalent at a given plot through time. And that would be very much similar to in the analyses tab. I guess this is maybe a better example. I might go to this, like I said, the Summit County Forest Health Task Force is a real project, but it's literally just getting started. So I might go to like a Trout Unlimited project real quick and look at some sites that have been monitored through time. And this is, I think, a West Virginia project. And if I look at its locations, once it loads up these pictures that volunteers took, if you go to your locations tab in your view data, this should load up all of the locations, and then you can sort the data by the number of observations at that location. So Boyd Run has been measured 25 times. So if imagine this being snow water equivalent at that fence, measured 25 times, you could view that data, and then you could get a dynamic real-time visualization of your variable, your snow water equivalents, through time. So what this is going to do is it's going to load up Boyd Run, and it's going to show a map, and it's going to show, in this case, air temperature through time. But it could just as well be snow water equivalents. In this case, they've also measured pH through time, and so you can look at the variability in pH through time. So if you measure multiple things, you can look at these trends as need be. It'd be interesting to look at connectivity, for example. So I think, Doug, that's a great question, and you could do just that. The question then becomes, when do you actually stop? But you could have it kind of an ongoing thing where every March you measure that variable at that location.
One more quick question. Can you download your data as a CSV and then plot multiple variables together?
You can. In fact, we're currently right now in the process of deploying a checkbox of check all. So you could actually look at air temp and pH on the same chart from a visualization standpoint. But from a downloading standpoint, if you go back to the project profile, for all projects, there's a way to do just that, and that's downloading data as a CSV. And when you do that, you click the download button, and it lets you actually look at all the data you've collected in your project and exclude certain things. So the default is to include all of the things that's been collected in this project over time, but if you don't want to download stream stage, you can just yank it. And so you're kind of customizing your download as Excel or CSV, and then you name your file as of March 5, 2015, and then there's your download. Great question.
Thank you.
You bet. Any other questions? One thing I didn't mention on my end kind of with is this invite members. How does people kind of, especially if you have a classroom setting, how do you invite those students to become members? This is a really nice, easy way to do that. If you're a teacher, you can enter Joey, whatever the first name is and last name, Surai or something, and then if you knew the student's email, you could enter their email, and what this will do is it'll automatically register them on sitsi.org as a first name dot last name username, and then it'll randomly generate a password for that student, and it'll send them an email, and then it'll also automatically make them a contributor to your project. So in one fell swoop, Joey then gets an email and can click an email and go straight to the project profile page, which is where they would then use your data sheet to enter data. The other way to join a project is, of course, manually. If you have a student or a community member who wants to actually be, as a member of the public, a citizen scientist, they can request to join your project at any time. And this will send an email to you as a coordinator to say that so-and-so actually wanted to join your project. And then you'd have to work with them to make sure they have the proper training, and you in fact approve them as a member of your project. So I think we're out of time. I think we're hitting right about 5 o'clock. Any other questions? I'm always available via email, gregory.newman@colostate.edu, to assist with any questions you might have, and